Hi, I'm Mai Yamakawa. I'm a neurologist and postdoc training in RexAc lab with bioinformatics. First, I'd like to thank the organizers for the opportunity for me to present today. I'm going to talk about a project titled Molecular Phenotyping of Rare Peripheral Immune Cells in Alzheimer's Disease Brain using single-cell genomics. Alzheimer's dementia is a multi-step disease with robust neuroinflammation. There are two characteristic pathological findings. First one is senile plaques, which is a deposition of amyloid beta. And then the second one is neurofibrillary tangles, which is a deposition of phosphorylated tau. And from cohort study of CSF analysis, we know that these pathological events occur almost 10 years or more apart and forming two steps in the pathological development. In between, we also know that the amyloid beta senile plaques are surrounded by reactive microglia. So we know that there is an immune component in the pathology development. And then recent advancement in population genetics revealed numerous immune-associated risk genes, including CD33, TREM2, and genes in MHC regions, major histocompatibility regions. And these genomics findings implicate the causal role of neuroinflammation in AD pathogenesis rather than just being reactive. So again, the AD pathology development is multi-step, and most of the focus in the microglia or neuroimmune studies are focused in the amyloid part. And microglia has been characterized with single-cell genomics, and we know that there are multiple states of microglia, including activated, amyloid-responsive, dystrophic, homeostatic, etc., etc. But we quite don't know yet what is happening in neuroimmune axis or microglia or else when it comes to the point that patients become cognitively impaired, as well as they start to have pathology with tau deposition. So here our key questions were, do neuroinflammation pathways differ between against amyloidopathy or early stage and tau-associated neurodegeneration, or in late stage where patients become symptomatic? And what are the peripheral immune cells involved in AD brain? And which cell types do they interact with in AD brain? And what are the key signaling molecules that could be therapeutic target? So the summary of the results, we looked at the microglia, and then we found that CD8 T cells are in the brain. And we performed a network module generation using WCNA. I'll get there. We identified two disease-associated modules for each cell types. And then we performed cell-cell interaction analysis and found some interesting, already literature-reported targets that could be further investigated. So there are multiple large consortium reporting and depositing the large single-cell RNA-seq datasets of the brain for Alzheimer's disease. We use Seattle Alzheimer's Disease Brain Cell Atlas, or now we call it CAD, which is comprised of 89 donors with detailed pathological evaluation. It includes single nuclear RNA-seq, ATAC-seq, and genotyping. And those samples came from middle temporal gyrus in the brain. And you can actually go onto their website and download the Surat object or data object for each cell types or all the data. And they also have a nice interactive cell viewer. And here is the UMAP of each cell type. And this gray thing in the middle, I think I can, laser pointer. Middle gray thing is microglia PVM. And there's this tiny subcluster in that population where it's positive for CD247, which marks both T cells and natural killer cells. And these cells are comprised of 800 cells coming from 79 donors. So for these data, cell types are actually already annotated by the consortium, but for lymphocyte annotation, we really closely looked at with the known markers, and then performed cell proportions analysis. And for molecular characterization, we decided to do HGWCNA, which is an extension of conventional WCNA, or weighted gene co-expression network analysis, which was designed to overcome some of the key difficulties with single-cell RNA-seq data. And after annotating those two cell types, we included all the other major cell types from the same data set. And those happened to be 23 shared donors. And we performed cell-cell interaction analysis using multi-niche net. Multi-niche net is another extension of NicheNet, which is unique of incorporating target gene expression predictability by the ligands. And multi-niche net uses pseudobulk as an input to perform the differential gene expression analysis. And the key component is to overcome multi-sample data set. And then it's also designed to compare multiple conditions. As for quality control, initially it started with 40,000 nuclei from 89 donors. Among them, 800 of cells were lymphocytes. And conventional QC was performed with percentage mitochondrial genes less than 5%. And then we decided to remove cases over 90 years old because of a higher incidence of co-occurrence of Lewy body pathology. We also removed neocortical Lewy body pathology. We thought it could be confounding. When we were clustering lymphocytes, we found one donor forming one distinct clusters. We decided to remove this donor from downstream analysis. And resulting input for the further analysis was for microglia, it was 11,000 nuclei from 28 donors. And for CD8 T cells, there were 384 cells from 70 donors. So we used different markers to mark those lymphocytes. We found natural killer cells, CD4 T cells, and CD8 T cells. Interestingly, the CD8 T cells were the only one significantly increased in dementia brain. We didn't see this increase in neither natural killer cells or CD4 T cells. These cells, after manual curation marker interpretation, were determined to be effector memory T cells with re-expression of CD45 RA or PTPRC, which is also known to be a brain-resident memory T cells. And these cells also are reported to be high in AD patients' CSF. Okay, so moving on to this network module generation, first starting with CD8 T cells. We found two network modules that were significantly associated with the disease status. And the HAP genes are the most highly connected to the network among those modules. And we can see that the immunomodulatory module on the right has multiple immune-related genes at the top. And we decided to call it immunomodulatory module. And the geo enrichment analysis implicated positive regulation of type 2 interferon production. For the cytotoxic modules, CD8T2 module, if we actually only look at the HAP genes, they are all ribosomal genes. And then we also saw a lot of mitochondrial genes, which happen to be also a case for differential gene expression analysis. But down the list, but not too insignificant, there are immune-related genes, including CCL5, CCL4, the cytokines and CD8 T cell receptors. And if you do pathway analysis on all those genes, we found that alpha beta T cell activation is on the list. So we decided to call it a cytotoxic module. For the microglial modules, again, there were 2 modules enriched in the dementia cases. And with literature reported markers, the first module was enriched with amyloid responsive microglial markers. And then the second module was associated with DGEs against both amyloid beta and p-tau. So we decided to call it degeneration associated module. And as you can see, CERAT score is a pathological score against senile plaque. And for the degeneration-associated module, you can see a clear positive correlation with the more advanced pathological stages. Whereas amyloid-responsive module, it's not quite clear. And then Bragg stage is another pathological scoring system for neurofibrillary tangle. Again, the second module has a clear positive correlation, whereas the first amyloid-responsive module doesn't have clear positive correlation. Some of the HAP genes included no marker genes and disease-associated genes, and pathway enrichment showed a HIF1 signaling pathway for the first module, and then the second module was a lot of it. But anyhow, we decided to call the first module as amyloid responsive module, and then the second module as degeneration associated module. Moving on to the cell-cell interaction analysis, we had interest in CD8 T-cell interactions. So we decided to count the ligand and receptor pairs and then just look at across the cell types. And we saw that, so this is all the microglial states, and these are homeostatic and dystrophic states, and the blue is normal, red is dementia. And you can see that these 2 cell types lose the cell-cell interaction. And instead, the other 3 types, amyloid responsive and degeneration-associated microglia, and then transition is kind of in between them, have more increased, significant increase in the cell-cell interaction with CD8 T cells. The other important factor is that overall we do see increase in dementia cases of all CCIs. And the output of this analysis is quite big, and we are still in the process of analyzing them. So I'm just picking one ligand receptor pairs up to just point out. So we found the CXCL16 and CXCR6 axis between microglia and CD8 T cells. And this happened to be actually already reported in the context of Alzheimer's disease and aging, as these molecules are pointed out in CSF samples from AD patients. And then also we know that this axis plays a key role in infection against West Nile virus. So we identified 2 distinct disease-associated gene networks that are differentially correlated with pathology for microglia. We determined that the majority of T cells in the AD dementia brains are T temera with positive correlation with advanced pathology. Degeneration-associated microglia have the greatest CD8 T cell interactions, further supporting potential role in neurodegeneration. And we identify cell subtype and condition-specific cell-cell interaction, providing new targets for validation and mechanistic study. In summary, blocking CD8 T cells in brain could be an effective therapeutic target to reduce neurodegeneration in AD, as demonstrated in mice and organoid models. Our findings from human AD dementia brain single-cell transcriptomic data further supports this, and clinical trials are needed to prove its efficacy. These are my acknowledgement. Thank you for listening, and I welcome any comments and questions.
Nice talk. For the CD8 T cells, it seems like you have a very limited number of them available from these data sets. Are there any other single cell data sets where you have TCR information that, I mean, CD8 might be there, but for them to be functional, they need to be clonally expanded, reacting to something, or you think just just the fact that there are some CD8s without antigen specificity is a driving force? Or?
That's a great question. Thank you for asking. So those cell types are effector memory cells, and in a progression of the CD8 T cell activation and maturation becoming memory cells, they're closer to the terminally differentiated T cells. But then along the line, farther than the temporal line, there's a subpopulation of T cells that becomes non-antigen presentation dependent. And in some contexts, it's called virtual memory T cells, that those cells are actually maintained by more of cytokines, not quite antigen presenting cells. And as far as I know, I'm not aware of any data set that looked at TCR, but there might be.
Thanks.